Okay, good evening, everybody. I make it 6 o'clock. So before we start, I'm just going to say a few words. So first of all, thank you so much for joining us this evening UK time, or afternoon or morning, depending on where you're based. And today, of course, is the 24th of March, so it's World TB Day. And but not only is it World TB Day, but it's World TB Day in the year 2021. So that means it's exactly 100 years since BCG was first used in human participants. So just a reminder before we go into today's talks that this event is being recorded and that the recording will be uploaded to YouTube and available from the Validate website. So Validate is an international network of researchers developing vaccines against globally significant diseases caused by complex intracellular pathogens. So we've been focusing for the past 3 years or so now on tuberculosis, leishmaniasis, melioidosis and leprosy. So this event is part of BCG 100, which is Validate's programme marking 100 years since the first use of BCG in humans. And you can find out more using the validatenetwork.org website, and there's going to be a link to the event and more details of the BCG 100 programme in the chat that you'll be able to see. So we're going to have 2 talks this evening, and then after the talks we're going to have some questions and answers. So you can ask questions in 2 ways. You can raise your hand at the end of the 2 talks, and then we will turn on your microphones and so that you can ask your question. It would be great if you could turn your camera on to ask the question as well. So you can also type in the chat if you prefer to do that, and then we will read out your question for the speakers on your behalf. I just want to remind you that if you are under the age of 18, please refrain from asking questions via the microphone and video, but you can ask questions via the chat. So today we are going to be hearing from Professor Paul Fine and Professor Helen McShane, and I couldn't think of 2 better people today to talk about BCG. So Professor Paul Fine is a Professor of Communicable Disease Epidemiology at the London School of Hygiene and Tropical Medicine, and Professor Helen McShane is the Validate Network Director of the Oxford NIHR Biomedical Research Centre, also a Professor of Vaccinology at Oxford University and the Deputy Head of Translation in Medical Sciences Division. So without any further ado, I would like to hand over to Paul Fine, who's going to give the first of the 2 talks that we're going to hear this evening. Thank you, Paul.
Thank you, Helen, and hello everybody out there. I think this program is called BCG Then and Now, and I meant to do the then part. An old story still surprises. Next slide, please.
Sorry, it's not advancing. Hang on. Go out of this mode. I don't know why that's not. Sorry about this. There, okay.
There we go. Okay, the old story part first, how it all began, and let's point out that BCG, we recognize, still being used. I'm sure that many people listening to this have actually received BCG vaccine. It's the oldest vaccine still in use today. Let's think about how it started. Next slide, please. BCG's origin. 3 gentlemen here, and they make me look a bit clean shaven. I think many of you know that BCG is a derivative of the bovine tubercle bacillus, Mycobacterium bovis. It was isolated by that man, Edmund Nocard, one of Pasteur's disciples. He was a veterinary surgeon. He got it out of the udder of a tuberculous cow in the year 1902, and he sent it to the Pasteur Institute in Lille, which is where Albert Calmette and Camille Guérin were based. And starting in 1908, they began serial passage of this isolate, ultimately did it 239 times in a medium which contained ox bile. I'm not sure about the ox bile, but that's always said in the books. And along the way, they repeatedly put this isolate back into calves to evaluate its pathogenicity. And they found that over time, its pathogenicity was lessened, it was attenuated. And so then, next slide, in the year 1921, 100 years ago today, it was first given to a human at that hospital there, Hôpital de la Charité in Paris, given by mouth to an infant whose mother died of tuberculosis shortly after delivery. The infant survived, thankfully, and so confidence arose in this bacillus as a vaccine. It was mass-produced at Lille, and by the year 1928, more than 114,000 infants had been vaccinated with it. But then something happened. Next slide. It happened in the city of Lübeck in Germany, where there was a program to vaccinate infants with BCG. And protocols were different then from what they are today, but in the same incubator in the city health department, they were growing their BCG bacilli, but they were also growing isolates from sputa Mycobacterium tuberculosis. And over one weekend, the labels apparently fell off the culture containers. A technician came in on Monday morning and reversed them, and 251 children received Mycobacterium tuberculosis per os rather than BCG, and 72 of them died. One of the great tragedies in the history of vaccines. And not surprisingly, confidence declined, almost disappeared. It said that Calmette died of a broken heart because of this. There was a court case, and they identified what had happened. And slowly, confidence came back, and the first trials, formal trials of BCG took place during the 1930s, ultimately leading to one of the best known trials carried out by the British MRC in 20,000 adolescents, which showed good effectiveness, and that led to wide introduction of the vaccine into Europe. WHO started introducing it in low and middle income countries, and in 1974, it was one of the 6 antigens included in the expanded program on immunization. And nowadays, we have a world where something like 85% of infants in the world receive BCG globally. And it's today very widely used, but it is still controversial, in large part because of its variable efficacy. Let me see the next slide, please. This just summarizes the history in the UK, where many of us are based right now. I mentioned the British MRC trial, and that led to the introduction of BCG into the UK in 1953 among tuberculin-negative adolescents. And I'm sure several people watching this received it in that way. In the 1960s, they added also neonatal BCG for high-risk populations in the UK. By the 1970s, because of the decline of tuberculosis in the country, first questions were raised about the overall cost benefit, whether it was worthwhile continuing vaccination routinely for all adolescents. As I say, that was discussed, but then HIV came along, and in the 1990s, there was caution expressed because of increase with HIV and multiple drug-resistant tuberculosis, and they thought, no, we aren't going to give up this vaccine now. But they ultimately did in 2005. BCG program shifted. They stopped the routine vaccination of tuberculosis-negative adolescents. BCG in this country now is given to high-risk populations, infants in districts where the incidence of tuberculosis is greater than 40 per 100,000, or who have parents or grandparents from high tuberculosis incidence parts of the world. Next slide. And here is a slide which is sometimes called the greatest headache in public health. This is one version of a slide which some of you may have seen in various formats. It shows the efficacy of BCG determined by controlled trials in that sort of blue color, or by observational studies in the yellow lines. The axis here is the protective efficacy. You see it goes from -50 to 0 up to +90. That's the percentage of recipients of the vaccine who are protected by it. And you notice that the results are all over the place. And that's the headache. Next slide, please. I circle here, I circle here just 3 of the studies. At the top, that's British school children. You notice it had about 78% efficacy. That was the MRC trial in the 1950s. But then you notice below Georgia, Alabama, and Georgia school children, studies carried out in the US where they showed no effectiveness whatsoever. In fact, Georgia school children, the point estimate was less than 0. It was about -20%, meaning that those who received the vaccine were more likely to get tuberculosis than those who didn't. So those were the results that came out during the 1950s, and they led to decades of argument. Next slide. And a variety of explanations were raised, and I just summarized them here. People looked at the trials and wondered about methodological differences in the way they were carried out. Were there genetic differences between the populations? Were there differences in Mycobacterium tuberculosis between the populations? Was it just differences in epidemiology in terms of the annual risk of infection or pathogenesis? Were there nutritional differences between BCG strains? Or that last one there, differences in environmental mycobacteria. I mentioned 50+ years of argument, but maybe now a consensus. I haven't taken a vote of people in the field now to say what explanation would they favor, but my guess is that a majority would now favor the last line there, italicized, that it has to do with differences in environmental mycobacteria to which we are all exposed and in different ways in different parts of the world. Those of you who are in the UK who took a shower last night, took a shower in an aerosol of Mycobacterium kansasi. It makes a biofilm in warm water pipes. These bacilli are all over the place, the environmentals. So let's look at the evidence supporting that. Next slide, please. One of them is here, the map of the United States, shaded according to the prevalence of sensitivity to Mycobacterium intracellulare, one of the common environmental mycobacteria, grows in soil and dust. These data come from testing 275,000 naval recruits. You see those 2 trials which found no effectiveness of BCG. They happen to have taken place in just those parts of the world where the prevalence of sensitivity is extremely high. Virtually all of those participants had been also exposed to and infected with this Mycobacterium intracellulare. Next slide. And here, these are data on more than 1 million US Navy recruits, the incidence of tuberculosis on the right there, as a function of their initial skin test sensitivity to tuberculin. That's sometimes called a Seibert antigen, that's S, or to another antigen, either to Mycobacterium intracellulare or Gaussa, that's the BG. And you notice that the incidence of TB was lowest, 29 per 100,000, among those in whom the sensitivity to the Bacillus intracellulare or Gaussa was greater than that to M. tuberculosis antigen, the Seibert antigen. So they had been infected with these environmental mycobacteria, and look, they had the lowest incidence of tuberculosis. So one of the explanations there explaining these differences in BCG's effectiveness around the world is that in many areas, people are exposed to other, i.e. environmental mycobacteria, which provide as much protection as BCG can. BCG doesn't add anything, so it comes out to have very low measurable effectiveness or efficacy if it's a trial. Next slide. Okay, here just some data from recent systematic review of BCG, the effectiveness against tuberculosis disease, pointing out that BCG is systematically more effective at higher than at lower latitudes. The top part of this slide, studies carried out at greater than 40 degrees latitude. At the bottom, studies at less than 20 degrees latitude from the equator. And you notice that overall it's about 50% effectiveness, but appreciably greater in that top group in the high latitude studies, and I pointed an arrow there to the MRC trial, than in the studies carried out closer to the equator, where many of them do not show significant protection at all. That reflects those results all over the place of the summary trial slide I showed earlier. So that's by latitude. Let's look at the next slide. This breaks down the various studies according to whether the BCG was given to immunologically naive individuals or to those who had prior mycobacterial sensitivity. And you see from this at the top, with the BCG to neonates or to school age with stringent tuberculin screen, as in the UK, because given to tuberculin negative adolescents, much higher effectiveness than in the studies toward the bottom, where it was given to various ages with no tuberculin screen, given independent of prior tuberculin status. So that's all consistent with the environmental mycobacteria exposure hypothesis explaining these differences in effectiveness. Next slide. More recently, there have been studies on the effectiveness of BCG in preventing infection with the tubercle bacillus using interferon gamma assays as a measure of infection. And we see once again considerable variation in studies carried out in various places, and overall about 15%, indicating a little protection against infection, not as great as the protection against disease in northern latitudes. Next slide. What about the overall impact of BCG around the world? I said that something like 85% of world's infants now receive BCG. In the center there, you see the coverage of BCG among infants in the world, which started in the 70s at about 15% and going up to what we see now by 85% in recent years. Over on the right, you see TB incidence over the last 20 years or so. BCG has not had a clear effect on that. Look over on the left, the other EPI vaccine, diphtheria, tetanus, pertussis, measles, polio. There you see the coverage portrayed in the same way as the coverage for BCG, and look at those incredible declines of all of those diseases. Clearly the vaccine's having market effects, but we don't see it with BCG. However, BCG effectiveness is consistently high against TB meningitis. Every study everywhere has shown that, and that's a major justification for WHO's continued recommendation that BCG be given in infancy, and it's so given in virtually all the low and middle income countries in the world. Next slide. And now to some of the surprises. Here we go. Next, leprosy. BCG and leprosy. Of course, the leprosy bacillus is also a mycobacterium. The first hint that BCG might be relevant for leprosy came from a study in Argentina. It was found that those who had received BCG vaccine were more likely to be sensitive to a skin test antigen used derived from the leprosy bacillus. It's known as the Matsuda reagent. BCG induces sensitivity to that. And that discussion of this ultimately led to the first randomized controlled trial in Uganda, carried out by the British MRC in the 1960s, which found very high effectiveness of BCG against leprosy there, about 80%. And subsequent trials in various countries, New Guinea, Burma, South India, Malawi, etc., effectiveness from 20% to 90%. If you look at those, they're actually greater than the protections levels against tuberculosis. It's a better vaccine against leprosy than against TB. And BCG, being so widely used, is certainly contributing to the global decline of leprosy around the world towards, the phrase they use is elimination as a public health problem. It's having a major impact there on leprosy. Next slide. BCG and bladder cancer. Well, how did this start? In the late 1920s, Raymond Pearl, a famous demographer, wrote a paper on cancer and tuberculosis. He did case control studies of cancer patients and found that his cancer patients were less likely to have had tuberculosis than his controls. And he wondered, what's going on here? Is somehow tubercle disease or infection protecting against cancer? It led to many studies over the next 30 years. And ultimately, someone decided to evaluate the effectiveness of BCG given by urethral catheter into the bladder for treatment of bladder cancer. And lo and behold, it was highly effective. And BCG is widely used for this purpose throughout the world now. In 1990, FDA, Food and Drug Administration, approved BCG for bladder cancer in the USA. That's the only approved use of BCG in the USA. It's not approved for prevention of tuberculosis. Next slide. What about bovine TB? That, of course, is the Mycobacterium bovis, the origin of BCG. That bacillus is responsible for something like 1% of tuberculosis in the UK now. It's found in particular among elderly patients who probably contracted the infection in the pre-pasteurization era. Pasteurization became routine in the UK in the 1950s. It's also found in cattle and in number of wildlife species, I list there, and in badgers, who provide a reservoir for Mycobacterium bovis infections in cattle in this country. Next slide. And here just a slide showing the increase in bovine TB in the UK from 1986 to 2000 to 2012. Incredible increase, which isn't really understood. And you see from the maps, it started in the southwest corner of the country and spread from that. A major problem in this country. Next slide. BCG vaccine to the rescue. You see this study, BCG vaccination reduces risk of tubercle infection in badgers. Given to badgers intramuscularly 10 times the human dose. And on the right there, how to run a scheme to vaccinate badgers. You can find that online now. Anybody who wants to know how to do it. Well, the first thing, you have to catch your badger. So it's an interesting challenge. So this is being used to some extent in this country now. Of course, BCG has also been used to prevent tubercle disease in cattle, but it's controversial because BCG induces tuberculin reactivity, which is the main screening tool for controlling bovine TB, and it makes cattle ineligible for export. And so that's problematic, and it's led to a lot of interest in DIVA assays, trying to distinguish infection from vaccination as the reason for tuberculin sensitivity. Next slide. Then there's this story of non-specific effects of BCG. Several studies have suggested BCG vaccination is associated with reduced all-cause mortality to an extent greater than be explained by TB. These are studies typically in children. Look at the next slide. There's a summary slide from independent systematic review by Higgins and colleagues, published in BMJ in 2016. And there, another forest plot. Those points to the left of that line reflect lower mortality among vaccinated than unvaccinated children. Virtually all of those studies. Now, this is controversial. Most of the studies are observational, and we all know that people who receive vaccines are different from those who don't receive vaccines in many populations. But I think there is a consensus out there. Again, I've taken no poll, but my impression is that there's increasing sense out there, consensus, that BCG is associated with beneficial non-specific effects of this sort. Next slide. And presto, what do we have here? BCG against COVID, the disease of the year. And there have been half a dozen trials out there. Of course, they were started months ago during the desperate years, months of COVID. Now we have with a variety of specific vaccines, which are doing very well. And so I think the interest in BCG against COVID has declined. Last slide, I think. So we can ask here whether this does indicate beneficial priming of the immune system by BCG. I'm not an immunologist. The next speaker may pick up on this, but BCG contains mycolic acids, which are major component of Freud's adjuvant, a very important immunological reagent. And maybe there's a link to the hygiene hypothesis. You know, this hypothesis that one reason why there's an increase in allergic disease in high income countries is that basically kids don't eat enough dirt. They aren't exposed to enough antigens. Yes. And maybe BCG is a useful antigen, broad antigen, to give to young people to protect them against a variety of things. So there's a broad look at the history and the surprises. Thanks very much.
Okay, thank you very much, Paul. That was fantastic. So just a reminder to put your questions in the chat. We're going to hear the next talk, and then we'll do questions for both talks at the end of that. You can, at the end of the talk, also raise your hand and put your video on and ask your questions in person as well. But yeah, thank you, Paul. Really fantastic talk. And it's amazing how much, we've been using BCG for 100 years, and yet there's still so much that we don't know about it. But the next talk, I think, may give us some insights into that, I hope. So Helen, Professor Helen McShane, it's a real pleasure to introduce Helen to give the next talk on BCG and beyond. Thank you.
Helen, can I just check you can see my slides and that they're in proper slide view?
I can see your slides, yes. Yeah,
And they're in slide view.
If people can't see the slides, if they can just put a note in their chat.
And you can hear me okay too?
I can hear you absolutely fine, yeah.
Super, thank you. Okay, well, Paul, after that fantastic tour de force over the last 100 years, it falls to me to do the now, in the then and now, and to really bring us up to date with where we are with TB vaccine research. And I thought I'd start really with this slide, which of course is a chest x-ray of someone with pulmonary TB. You can see the cavitation there and the pneumonia, the consolidation in the right upper lobe. This white white chunk at the top of the chest. And I think really the point here, and Paul illustrated it very beautifully, is despite 100 years of BCG, and despite 70 years since the British MRC study, there are still 10 million new cases of TB every single year throughout the world, and 1.4 million deaths throughout the world. And whilst for the last 12 months at least, the world has been rather distracted by a very nasty virus, the truth of it is that TB has killed 1.4 million people or more every single year. TB is globally the most important infectious cause of death. And this is on a background, as Paul said, of a sort of approximately 85% BCG vaccine coverage. So clearly, whatever BCG does do, it doesn't do enough. Paul also mentioned that one of the reasons that all of the strategies to develop a new TB vaccine regimen include BCG in some shape or form is because BCG in all the trials, when given at birth, gives consistent and reliable protection against TB meningitis, which is a form of disseminated TB, and that's what this chest x-ray illustrates, where TB is spread outside of the lung and into, in the case of meningitis, into the brain. And that's particularly common in infants and young children. And here you can see a chest x-ray of disseminated TB, where you can see, unlike the last picture, where apart from that rather large white opacity on the top left of your screen, the rest of the lung fields are rather clear, and that black means that there's air going in and out. Whereas you can see in this chest x-ray, there's no significant area of black, and really there's a sort of rather what we call miliary dotted pattern of white throughout those lung fields. This patient, I think it's fair to assume, is pretty sick. And just to show as a tease, really, this last slide, which is one I like showing to medical students to get them to guess what it illustrates. This is what we're trying to prevent with a vaccine, which is TB disease that we have to do surgical intervention for. So you'll see these very symmetrical, I don't think my mouse is working. Okay, you'll see the very obvious symmetrical circles in the top left of your screen, the top right of this chest x-ray. And what this is something called plombage. And this was a surgical treatment for TB, where essentially polystyrene plastic balls were inserted between the two layers of pleura covering the lungs to compress the lung in order to kill the TB. Because we know that TB is aerophilic, that means it likes oxygen, and if TB doesn't grow without oxygen. So if you compress the lung and stop oxygen getting into that part of the lung, then you can kill TB. But of course, we'd rather do that by preventing TB in the first place with a vaccine. So the global TB burden in 2020, or this is 2020, but the figures won't be very different this year, is that we have 10 million new cases and 1.4 million deaths. That is 100 years after BCG. And clearly there is a very urgent need to develop new tools. We've seen this year the speed with which we can develop new tools against a pathogen 13 months ago no one had heard of. But it's really salutary to note that TB is a pathogen that's been around since the pharaohs, and still this year, and SARS-CoV-2 aside, kills more people than any other infectious disease. We have a global plan to end TB, which I have to say as 2022 looks pretty close. I think looks increasingly ambitious. It is important to set ambitious targets and to develop a roadmap of how we can develop new tools, how we can develop new diagnostic tests, how we can develop new antibiotics to treat TB. But ultimately what we really need to get on top of this disease, and we've seen this of course with COVID and the power of vaccination, is a better vaccine. So vaccination, I think, I suspect I'm speaking to the converted from many people listening, is the most cost-effective health intervention available to us. We've eradicated smallpox with the only infectious disease ever eradicated was with vaccination. We have come tantalizingly close to eradicating polio. We have 27 vaccines currently licensed at the last count, and of course the most recent of those was until recently, HPV vaccine for human papillomavirus to prevent cervical cancer. But of course, we now have vaccines in the press every day with the exciting news that we have, as Paul said, very specific, highly effective vaccines, multiple vaccines against SARS-CoV-2. So we have the WHO expanded program on immunization, routine immunization program for infants throughout the world, and up to 95% of infants globally are vaccinated. And as Paul said, BCG is a very well established part of that EPI program, given the WHO recommendations are that BCG is given at birth or as close to birth as possible, because the evidence, and in part this links with the preventing interference with non-tuberculous mycobacteria, the best evidence that BCG protects is if you give it as soon as possible after birth. And it's estimated, and I think this is a highly conservative estimate, that about 2.5 million lives are saved every year by that vaccination program. So some of the things we have to think about if we're developing a new vaccine is who would we give it to? And traditionally, when one thinks about what are called target product profiles, is we think about this in terms of age and of risk factors. So we know that infants throughout the world, particularly in low and middle income settings, have a huge burden of TB disease, particularly and most obviously in sub-Saharan Africa, where the epicenter of TB is. So there's no doubt at all that infants, if we had a more effective TB vaccine, infants, particularly in low and middle income settings, would be eligible and would benefit from that vaccine. We also know that adolescents and young adults also have a high risk of TB disease. Now, although their risk of TB disease is not as high as those infants, there are two reasons why targeting adolescents and young adults is particularly attractive. Firstly, because that's where the greatest economic impact of TB is. TB hits people at their most economically productive. Young adults who otherwise would be going out to work, develop TB, can be chronically sick for months and years. And this is in part why it contributes to the challenge in an LMIC setting of moving out of the cycles of poverty that we see there. It's also the case that adolescents and young adults are the main population responsible for transmission. So adolescents and young adults develop what we call smear-positive TB, which is where one of those cavities, or that pneumonia I showed you on the first slide, erodes into an airway, one of the major airways, and then when that patient coughs, they cough up quite a lot of infectious particles that it's very easy then to transmit. We know that infants don't develop smear-positive TB, so although adolescents have a lower rate of TB than infants, actually if you could stop transmission by vaccinating those adolescents, you'd have a bigger effect on reducing TB incidence. And we know that HIV-infected adults remain at increased risk of TB despite widespread use of antiretroviral therapy. We knew in the pre-antiretroviral era that TB was a very significant risk factor for people with HIV, and unfortunately that risk hasn't gone away completely despite the fact that there's widespread global use of antiretroviral therapy now. The other way to think about who we might vaccinate with a TB vaccine is what path in the infection and disease cycle are we trying to intervene in here. So most of the vaccines that we give are given prophylactically. So they're given before someone is infected in order to stop them becoming infected. And if we stop people becoming infected, we stop them developing disease. And that really means giving a vaccine to infants, or it means giving a vaccine to people who are not latently infected. And that's either uninfected adolescents or young adults. But given that in South Africa, which is where I've spent a large part of the last 20 years working, we estimate that over 90% of young adults are already latently infected with TB, then giving a vaccine to prevent that infection seems rather worthless because they're already infected. So in that context, what we would try and do is give what we call a post-exposure vaccine to someone who is already infected, to try and see if that vaccination would either eliminate or help them clear that infection, or at the very least would prevent that latent or dormant infection going on to active disease. So they would stay well, even if they remained infected. And then the other population of people that we might think about giving a vaccine to are people who've already got TB disease. And the reason for that is that if you have TB disease, unlike if you have normal pneumonia, it might take you 7 days or 10 days of antibiotics. If you have TB, it can take 6 months. The standard course of treatment is for 6 months of antibiotics. And we know that if you had drug-resistant TB, particularly multi-drug resistant or extensively drug-resistant TB, you sometimes need to take treatment for up to 2 years. So if we had a vaccine that you could give at the same time as giving antibiotics as an adjunct to chemotherapy, then perhaps we could use that vaccine to shorten the period of time we need to give antibiotics for. And that would help stop the development of further resistance, because we know if people have to take tablets for 6 months, let alone 2 years, then when they start to feel a bit better, they might take their tablets less reliably. They might forget to take tablets. It's pretty difficult to remember to take tablets 3 times a day for 6 months or 2 years. And that, of course, then creates the perfect conditions to develop resistance. So those are the kind of populations we might think about giving a new TB vaccine to. But we need to now develop a new TB vaccine. And this slide is deliberately blank because this slide shows the clinical development pipeline in the year 2000. So 21 years ago, this slide illustrates the TB vaccine candidates that were being tested in the clinic. There weren't any. And that compares with about 50 vaccines at the same time being tested in the clinic for malaria and about the same number for HIV. And you've seen this year just how quickly, when funding is not rate limiting, how quickly we can develop vaccines. I'm very happy to tell you that the situation in October of last year, and of course now as well, is very different. So in the last 20 years, we've now moved globally as a field about 14 candidate vaccines into clinical trials. You can see here the preclinical panel on the far left are vaccines that are still being tested in animals, but then you can see that we've moved vaccines through into phase 1, phase 2, and some into phase 3 trials. So it's great. We now have a pipeline of vaccines that have moved forward. But TB, unfortunately, isn't as easy to develop a vaccine against as SARS-CoV-2. And this is going to be a very long haul. And what we need is sustained investment and sustained resources, sustained research, if we're to realise the potential of this pipeline of vaccines that we now have. I'd like to now show you 2 slides which are the really sort of hot off the, well, they're not that hot off the press anymore, but the most exciting data to date in the field of TB vaccines. And I think both of these papers came out in the last 2 years, and they really do show us some hope that it might be possible to develop a better TB vaccine. So the first, and this was work conducted in South Africa with many collaborators of mine, the South African TB Vaccine Initiative, and then Eliza Neames, who is the first author here on this New England Journal paper, showed that boosting BCG with BCG can reduce TB infection. So the endpoint in this trial was not TB disease, it was TB infection, asymptomatic infection. And in this trial, they took 990 BCG vaccinated, TB uninfected adolescents in South Africa. They were randomized to get a BCG booster, either in adolescents, or to get H4 IC31, which is an experimental vaccine, or placebo. And the endpoint in the study, as I've said, is TB infection, and the primary that was the primary endpoint. And the secondary endpoint was sustained TB infection, so long-lasting TB infection. And you can see there in the panel that although there was no significant effect on overall TB infection, with that p-value being not significant, when you looked at cases of sustained TB infection, there were 6.7% of cases in the BCG revaccination arm, compared with 11.6% in the placebo arm, and that gave us a very significant p-value of 0.03. So this is really exciting because it does, for the first time, give us some data that in a particular area of the world, and there's a caveat to that, this may not work everywhere, and Paul and I could discuss that a lot, it may be that giving BCG more than once might help. The Western Cape is an area where there isn't much exposure to non-tuberculous mycobacteria, and that might be relevant, and that might be why there's a signal here. There's now big plans to repeat this study with larger numbers to see if this effect is real and to see how long lasting it is. But this was the first real demonstration that we could do better than BCG in humans. And you know, this was in 2018, so this was 97 years after BCG was first tested. That's how long it takes in TB. This second study, also published in the New England Journal, and this was conducted by Derek Tate and colleagues, and this is looking at the GSK GlaxoSmithKline vaccine, M72AS01. So this is a protein adjuvant vaccine designed to boost the effects of BCG. What they did in this study was took TB subjects in South Africa again, who were already latently infected with TB. These were all adults, and they were boosted with M72AS01. And the endpoint here was TB disease, and it was 3 years of follow-up, and they had an enormously exciting vaccine efficacy of 49.7%. So you can see in this figure, you can see that the placebo arm, this is a diagram which shows each step down is the number of cases. And you can see that as the trial goes on, there's more and more separation between that orange line, the M72 line, where there are less cases, and the blue line, the placebo line, where there are more cases. So again, an enormously exciting result. There are a lot of questions still to ask. Would it work in people who are not latently infected? We don't know because they weren't tested in this trial. And the complexity of actually scaling up this vaccine and testing it again is are not insignificant. But I think, again, really important proof of concept that we can do something and we can develop vaccines that work better than BCG to prevent TB in what is considered the epicenter of the TB epidemic in South Africa. So 3 slides now, just showing you the next generation vaccines. So this is really about making BCG better. So this is looking at a candidate called VPM1002, which is a recombinant BCG. This is a genetically engineered BCG. And this was the first data released from this vaccine. This is a mouse study. And if you look here, the y-axis here is the amount of TB basically in the lungs of the animals. And you can see that the naive group at the top have the most TB, the BCG group are in the middle, and the RBCG group, the recombinant BCG group, are the lowest, with stars indicating statistical significance. So evidence in this mouse study, and there's data in other animal studies, and this vaccine is now being tested in a human efficacy trial, that this vaccine is better than the normal BCG developed 100 years ago. Making BCG better too? Well, as Paul said, BCG is attenuated Mycobacterium bovis, and there is a school of thought that actually that's the wrong strain of Mycobacteria. We should be rationally attenuating Mycobacterium tuberculosis, which causes most of the human disease in the world. So Carlos Martin and his colleagues in Zaragoza took TB and rationally attenuated it, and have developed a vaccine candidate called MTBVAC, which is a rationally attenuated strain of TB. And you can see here, this is a human clinical trial, and this shows the ELISpot responses. So we're looking here at levels of T cells induced by this vaccine. And if you look at the two right panels, you can see BCG there on the far right, and then MTB-VAP at 5 × 10⁵. You can see they're inducing pretty comparable responses. So this vaccine, which is in animals at least safer than BCG, may be a better candidate than BCG going forward. And again, that is being tested in efficacy trials. And then finally, and this might feel a bit wild, so bear with me, there's now data from 3 different studies. The first one done about 70 years ago, and then the two most recently, done much more recently, that giving BCG intravenously, injecting it into the vein, is significantly better than giving it into the skin intradermally in non-human primates. And this is work that Sally Sharp has done and has most recently been replicated by Bob Cedar and his group at the NIH. And you can see here, so the score on the vertical axis is the amount of pathology, or how much disease there is in the animal. And you can see that the green box there is the naive animals, the blue box is intradermal, the sort of pink box, I hope I'm describing these colours helpfully, is intradermal followed by intratracheal, and then that purple box on the far right is intravenous. And you can see pretty clearly that intravenous BCG is much better than the other routes. Now, I am not proposing that intravenous BCG is a deployable vaccine to be given to neonates throughout the developing world. I don't think that's feasible or the right way forward. But I do think that this result is telling us something. And I think if we could do some careful studies in humans and in animals to try and understand what it is that's making intravenous BCG so good, then we could try and mimic that with a deployable vaccine strategy. And then just to finish, a couple of slides on some work we've been doing in my group in Oxford. There is some animal data to suggest that giving a TB vaccine into the lungs is the best way to protect. And that sort of makes sense because that's how TB enters the body, through the lungs. So we're mimicking here the route of immunisation with the route of infection. And we know that BCG doesn't reliably protect against lung disease, and perhaps that's because we're not inducing a strong enough immune response at the site of infection. We give lots of drugs into the lung nowadays, and there's very standardized delivery systems to do this. So I think, and there are lots of logistic advantages to this. It's a needle-free approach, it's pain-free, and will be very suitable for mass immunization campaigns. So we've been doing some studies, experimental medicine studies in my group in Oxford, looking at can we give inhaled TB vaccines, both 2 candidate vaccines we've developed there, MVA-85A and ChAdOx1-85A, but also inhaled BCG using this little jet nebulizer I've shown a picture of here. And what we do is we then look at blood immune responses, and we take washings out of the lung to evaluate what the immune response is in the lung as well as in the blood. And these studies to date have been very successful. We've shown that inhaled vaccines, the ones we've tested, are safe, and that they stimulate very strong immune responses in the lungs. So we're interested in taking that forward to the next step now. So I hope after Paul's fabulous introduction, I've given you a sense of optimism that after what I would describe as decades of neglect in TB vaccine research, we have made a very significant progress over the last 20 years. It's important to remember that all of the strategies build on the current BCG vaccine because we don't want to lose that protection against TB meningitis, so critical in infants, and against which BCG is so effective. We can learn a lot from the trials that we do, whether or not the vaccines work. We do need better ways of testing these vaccines before we get to what are very big, very expensive, very time-consuming efficacy trials. There's no doubt at all that if we had validated animal models and if we had immune correlates of protection, the field would be transformed and vaccine development in TB would be considerably easier. But at the moment, what we're left with is there's no substitute for human efficacy testing. Helen, thank you.
Thank you, Helen. That's fantastic. And yeah, I mean, I strongly agree. There's no substitute for human efficacy testing. And I think, you know, this last year has shown us that actually there's no excuse really. I mean, we have this BCG vaccine. These studies show us that we can use it more effectively, perhaps. and protect more people, both through revaccination, through those different routes of delivery, as you say. So important to reflect on BCG, but actually I think we should accelerate going forward. And there's no excuse to take another 100 years to find a better way to use the BCG vaccine.
Absolutely.
So we have a couple of questions in the chat, but also I'd like to invite other people to post questions in the chat, or you can raise your hand if you would like to ask a question in person. When you raise your hand and I invite you to ask your question, Blakely will unmute your microphone, and so that might take a few moments. So we might have a little bit of a pause between when I've invited you to ask your question and when you can actually speak. So bear with us with the gap. So whilst you're all planning your questions, I might start off by going through some of the ones that we've had in the chat. And so the first question, Paul, is directed at you, and it's regarding the original MRC trial. So that particular trial had 2 arms or 3 arms to it, placebo arm, but also two different intervention arms. One was BCG and the other was Mycobacterium microti. Both microti and BCG look to be effective. Do you know why at that point BCG was chosen over microti for a vaccine to be used in the UK?
That's right. Mycobacterium microti was used in the trial, and it appeared to be virtually as effective as BCG. Why they decided on BCG, I'm sure there was a good deal of discussion at the time, and I really don't know. You know, their BCG had been around for a good deal, was known about it, and it was being increasingly used. So I think that was probably the main reason. There was a lot of experience with it, and the microti didn't look better, but it was providing somewhat similar protection. My guess is that's the reason why they're stuck with the BCG, familiarity. That's my guess.
I think that's probably right, Paul. I guess the other thing I wonder is if they had manufacturing, knowing that all problems are to do with manufacturing, if they had manufacturing processes already for BCG, yeah, it was just the easiest path forward, wasn't it? And I think the data was that microti and BCG gave the same result, so it wasn't that microti was better. It's a good question, though.
Yeah, and absolutely, you know, and again, I think the fact being able to manufacture a vaccine and having, you know, that sort of decades worth of history of safety data and use worldwide, you could see how that would tip the balance to BCG. But it would have been great to have, you know, it would be really great if somebody could find a historical document of the minutes of that meeting where that decision was taken. It would be a lovely thing to have. So another question from Camilo is, does BCG share more antigens with M. leprae than M. tuberculosis? And I hope that my panelists won't mind if I sort of chip in with some of the answers.
Please do, Helen, absolutely.
Please rescue me on that one. And anyway, I'm pretty sure that there's greater similarity with M. tuberculosis than with M. leprae, but you'll know the facts.
Yeah, no, you're right, Paul, that there is more similarity. I mean, Mycobacterium leprae, which you know far more than I do, but it's a really fascinating organism, and it's got a very pared down genome. So it has, you know, relatively few antigens compared to M. tuberculosis. I think maybe a third of the size, you know, the size of the expressed antigens. So whereas BCG and M. tuberculosis may differ in, you know, a few antigens in a small percentage, leprae is a really pared down version. So my suspicion would be that leprae is just less able to avoid the immune response induced by the BCG vaccine because it has, less flexibility in its genome to be able to escape that. But that's would be my view on that. But certainly in terms of antigen similarity, then there's no evidence that Lepre is more similar to BCG. I think it's just less able to avoid immunity. But Helen, what's your thoughts on that?
Completely agree. I was going to say exactly the same thing. I'm sure that if one lined up the genomes, I mean, TB and BCG are closer than BCG and Lepre for sure.
Okay, thank you. And so we have another question in the chat from Rita Gossin. And she's asking, she says there's one additional surprise of BCG that's come about in the last year is a study related to BCG vaccine use in type 1 diabetes patients to improve their glycemia levels. Has there been any follow-on studies from that? Do we know if that was replicated? Is that a thing, BCG in diabetes?
So I know the study that Rita is referring to. It's a very small study. I think there was an enormous amount of excitement from the diabetic community when it came out, but I think it was a very small, my recollection was not controlled study. I think, I think we'll do better to develop specific treatments for diabetes rather than think that BCG is a panacea that's going to prevent diabetes.
Paul, what's your thoughts on this one?
Well, I don't know this study, but the BCG has been tried again. I mentioned the history of studies of BCG in cancers, and there are quite a few studies out there and claims made. For example, BCG in treating breast cancer, all sorts of things. Most of them poor, small. At any rate, there's been a lot of enthusiasm about BCG over the years, and anyway, it's interesting. This issue of the non-specific effects, that's attracted most interest over the last few years, and I think many of us think there's something there. There's something there. Another trial carried out in Uganda was just published a couple of weeks ago.
Yes.
On BCG in infancy. This was the outcome here, was not TB, was not mortality, was just morbidity, a variety of illnesses, and indication of some protection for a period of 10 weeks or something in early infants. Just another result out there.
That was a very nice study, actually, and a very well conducted study as well from the UBRI unit. Yes, absolutely.
And it's known, for example, if you give BCG along with Hep B vaccine, you get a boosted immune response to the Hep B vaccine. It's acting as an adjuvant.
It's an adjuvant, exactly.
Yes,
exactly.
It's an adjuvant.
Exactly.
Yeah, absolutely. Thank you. So we have a question from Nick Wilcox, who is asking about BCG in people who are HIV positive. So can BCG protect people who are HIV positive from developing TB or from getting more severe disease?
Well, one of the challenges is a good question, Nick. One of the challenges is that, of course, BCG is contraindicated in people who are HIV positive because it's a live replicating vaccine. So WHO guidance, and indeed that's the reason why in HIV high burden areas, WHO guidance is now that the HIV status of the infant is known before they receive BCG vaccination, and that's because of the risk of disseminated BCG disease.
Helen, I meant, do people who've had BCG as children, are they protected when they get HIV later on?
No, they're not. No, they're not. So
are they protected against TB when they get HIV later on?
No, it doesn't.
No.
So
Okay.
Most adults who get HIV have had BCG vaccination as an infant, and it doesn't stop them getting TB. No.
Thank you.
No
Okay,
worries.
thank you, Nick. So there are some more questions in the chat, but I'm going to give people who have put their hand up a chance to ask their own questions to you directly. So our first question will be from Anshu Dahal.
You should be able to, Anshu, you should be able to, you figured it out.
Yes.
Hi. Are you hearing me?
Hi,
Yes, we can hear you.
perfectly.
Yeah.
Yes, I am from Nepal actually, and I'm currently in Germany. I'm fortunate to be attending this webinar actually. I have this question, like as a child, I was vaccinated with BCG, but I came up with a question that is the BCG only mandatory in poorer region of the world and not in the rich countries, suppose Europe or America? One is that, and another is if you get vaccinated with BCG vaccine, does it give protection against COVID infection? Thank you.
Maybe I can pick up on that.
All go for it.
Okay. BCG, you said mandatory in poorer countries in the world. BCG was included among the 6 antigens of EPI when EPI started in 1974. And, you know, EPI is the WHO's basic immunization regimen for the world. And during the 1970s or so, 80s, the vast majority of countries in the world recommended BCG. There were a couple of exceptions. One of them was Netherlands. One of them was the United States. The United States never recommended BCG because of the trials that I showed you. The United States Public Health Service carried out major trials during the 1950s and found no protection whatsoever. And so when some people ask me, why does the UK use BCG and the United States not? My answer is because both countries behaved responsibly. In both countries, they tried it. The US found it didn't work. They would have been irresponsible to give it. In the UK, it did work. They would have been irresponsible not to give it. So the vast majority, the Netherlands never gave it. That's another country that never gave it. Now, many of the Northern European countries have given up BCG as a routine over the last 20 years. I mentioned that in 2005, the UK stopped the routine BCG recommendation to all tuberculin-negative adolescents. It's now recommended only to high-risk groups. And that's just on the basis of cost effectiveness. The risk of TB in the general population in the UK now is very, very small, very small. And they found there's no need for it. 70% of the TB in the UK now is in people born abroad. And so it's targeted to those populations. It's the same in, I think, most of the Northern European countries now.
Yeah, perfect answer, Paul. Agree with that completely.
Thank you for that. So our next question, which is another one to be spoken question, is from Colin Larkworthy. Colin, if you'd like to ask your question, if you can unmute yourself, yeah.
Brilliant. Hi, Helen. Just a quick question for you both, really. I hope you can hear me to start with. When you've said about obviously intradermal, intravenous, obviously, and the nasal, do you have a point with, if, for example, there's a fear of needles, be it going intravenously, that there would be a case for many different routes to help tuberculosis and BCG and such?
So, Colin, I think the question, let me just check the question, was about the different routes.
Correct, yes.
And whether, I mean, I think We need to do much more work on looking at the different routes. There is animal data to show that both aerosol and intravenous BCG is better than intradermal BCG, but there isn't yet human data on those routes. I think we need to do some human clinical trials on this. I don't think it's going to be feasible to give BCG throughout the world intravenously to neonates in the developing world. But I do think that result, which has now been shown 3 times, does give us a model to help us understand what kind of immune response it's inducing, so then we can develop safer, more practical ways of doing that. So it's a good question.
I just might add one other thing there. I mentioned that BCG was given initially per os, was given orally. Yes, and that continued in some countries up until the 1950s. However, that was stopped because it was leading to glandular BCGiosis in a small proportion of recipients. So I think no countries now give it orally. It's given intradermally universally now.
Yes. And that's a good point, Paul. And of course, you know, these other routes, they may be more effective, but they may give us different side effects as well. So we would have, because there are usually a very small percentage of people who do develop BCG disease because of the BCG vaccine, because it is a live replicating vaccine.
Yeah. Thank you very much, guys.
Pleasure.
Thank you. So the next question we have for a live question is from a Dr. Nolan. If you'd like to unmute yourself and ask your question, please.
Hi, thank you. Really interesting talks. I'm Margaret Nolan. I'm at Stellenbosch University.
Hi.
So we know, hi. So we know we have various strains of BCG, and this impacts everything from virulence to immunogenicity, viability, and that it also has distinct protective effects then, depending on that. What are your thoughts on the impacts then of the use of these different BCG strains in research for comparative purposes? With one study using BCG Thai, someone else using a Pasteur strain, and the potential impacts of that and our ability to actually compare across different research studies?
Yeah, it's a good question actually, and we may have differing opinions on it, but I would suggest Paul, who is the oracle on BCG strains, starts with that, and then Helen and I can chip in with our thoughts.
I'm not the oracle on BCG strains. This has been discussed for decades. It's one of the explanations which has been put out there. There have been a number of studies which use different strains of BCG, for example, in the South India BCG trial, and found no difference between them. So it's recognized that some BCG vaccines are more reactogenic, are more likely to give a larger ulcer, give local lymphadenitis, but my sense is that the consensus, and again I haven't had a vote out there, the consensus has been that strain differences, genetic differences in BCG don't explain those differences that I showed you in the systematic reviews of BCG. That's been a conclusion of most of the systematic reviews at any rate. I know that there are some people who still raise questions about this, and I turn to the two Helens for wisdom on that one.
So I would agree with you completely, Paul. I think there's quite a lot. So people who do work in mice and in guinea pigs have done head-to-head comparisons of the different strains of BCG and shown no difference between them. And I think you're absolutely right the way you summarize the clinical evidence. I think with the one exception, I think possibly Tice is a BCG strain that seems to be less immunogenic in humans and may well be slightly less potent. But I think in general, whilst there's no doubt at all that there are genetic sequence difference between the different strains of BCG, I remain unconvinced that those sequence differences are important in terms of the efficacy difference we see.
Yeah, and I'll add my thoughts. I agree very much with Helen and Paul that there doesn't seem to be any evidence epidemiologically of a difference in efficacy by BCG strain, and also in the laboratory there, you can see some minor variation in your experimental result, according to the different BCG strain you use, but often that can be due to, a difference in the growth kinetic, rather than, one BCG strain will grow faster than another. So in your experimental model, because these are only models, you may see, a sort of slightly higher titer of immunogenicity or slightly more rapid onset of response, but they're minor and they don't impact your results. I think the related question, which is probably more important biologically, is what difference do you see in your experimental models according to what strain of M. tuberculosis you're challenging with, because we know that we do see some variation in the way that clinical M. tuberculosis strains behave. Mycobacterium africanum, for example, might be sort of quite different in many ways to quite an ancient M. tuberculosis strain compared to the modern strains, and then the Beijing strains again might be quite different. And obviously, if you're using a multi-drug resistant strain, then that might behave in a slightly different way. So if there's a gap in the way that we're using our animal models, I think it's more to do with our tendency to stick with the laboratory form of H37RV. Or Erdmann for our challenge models and not look at the clinical strains. But that's my personal opinion, and happy to throw that one back to Helen and Paul for their thoughts.
No, I think it's absolutely right, Helen. I think there are also differences in the way that the strains are manufactured, the way the strains are grown, which means that this is not comparing like for like. Some of the trials are done using lyophilized BCG. Some of the earlier trials were done using mid-log phase material. The problem is that it's not comparing like with like, and in such complex systems, I think there is inadequate data that BCG is a significant factor in the variability. Would be my summary.
Wonderful, thank you. That was, thank you very much.
Thank you.
There's a really good question in the chat here, Helen, from Simon. I don't know if you've seen it, Simon Mendelsohn at SATV, which maybe we should just talk about because it's really important, which is the tests for TB infection, QFT and TST, indicate prior exposure and not necessarily ongoing infection, and he's questioning the utility of that as a trial endpoint, which I think is an absolutely superb question. So to be clear, and I'll start and then hand over to Paul and Helen, in the trial where they used quantiferon conversion as an endpoint, the subjects were quantiferon negative at enrollment, so therefore they were looking at quantiferon conversion. But the problem with that is a lot of people who quantiferon convert also then revert. So that was where the sustained quantiferon conversion came in, because a lot of the original converters reverted to a negative quantiferon. And the other problem with all of this is that it's, in a sense, it's the oldest question in immunology. What the quantiferon is measuring is a T cell response to two relatively MTB specific antigens, eSTAT6 and CFP10. They're not entirely TB specific. They're in Kansasii, they're in Marinum, they are in some strains of NTM. And, we're measuring something really quite indirect from TB here. We're measuring the host immune response to it. So, and the question, we know that after patients have had TB treatment, some people keep their quantiferon positivity, some people lose it. So, what determines whether you maintain that T cell response, and is that T cell response dependent on persistent antigen? We don't know the answer to that. So you're absolutely right. It's a surrogate because it makes the trials smaller, quicker, cheaper, but we don't yet know whether it's a reliable surrogate for what we really want to protect against in these trials, which is disease. Helen, Paul.
I can't better that. That's fine.
Yeah, and I think I agree, and I suppose I'll kind of answer the question from the other end of the spectrum. So, you know, Helen was saying, you know, perhaps quantiferon is only picking up transient exposure and people aren't truly infected, and so it's problematic when you're using it as an endpoint for a clinical trial. But, you know, at the other end of the spectrum, if you're immunizing people who are TST positive or quantiferon positive, then you may well be including those with subclinical disease, with you know, minimal or subclinical disease. And certainly, you know, looking at the results of the recent population surveys in South Africa, there seems to be quite a high rate of prevalent tuberculosis. I think up to 50% of tuberculosis people don't have symptoms, they don't have a cough, they might not even have bacterial culture, yet if you do a chest x-ray you can see that they have TB, and obviously they'd have a positive TST response, quantiferon response. And so we're potentially recruiting people into our clinical vaccine trials who already have disease, and it's just we don't know that because we're just measuring the quantiferon response, and we don't. I mean, Helen, I don't know of any clinical trials that would do chest X-rays at recruitment into a trial. I mean, except for the latent TB trial that you did with MV85A when we first went to South Africa, when CT scans were done at baseline, but. I mean, it's not typically done, and certainly
not in large trials.
No, you couldn't.
No, exactly.
Yeah, so I think there the problem with that is, you know, your so if you look at, you know, the BCG revaccination study that Laura Rodriguez did in Brazil, you know, there was no efficacy of BCG revaccination in that study, but that was based on TST positive, so it could well have included people who already had, you know, minimal infection, you know, minimal disease and subclinical disease, plus those people who were transiently exposed at the end Helen was talking about. So it's really messy. It's a really messy inclusion criteria for you know, trying to define the effectiveness of your vaccine. And it's also a problem for safety. I think we've been actually incredibly lucky in TB vaccine development. I mean, as Helen said, you know, in the year 2020, there were no vaccines in the clinical development pipeline for TB, you know, beyond BCG. Now, you know, we're in a completely different place, and it's hugely exciting. But I think actually we've been really lucky. We haven't had any really severe adverse events associated with vaccination, because if we're using quantiferon positivity to recruit into trials, we may well be including some people who are already on the disease pathway. So I think, you know, as vaccine developers, we need to, you know, really pay attention to subclinical TB disease. And I think it'd be very difficult now if you were doing a clinical trial in a quantiferon positive population to not do X-ray screening, at least in a safety study, you know, in a sort of phase 1, phase 2 setting, to, you know, to understand how your vaccine, you know, may be performing, and the safety characteristics in that population.
No, I think that's absolutely right, Helen. I think the other issue, of course, is that you're not going to see a vaccine can't prevent someone from getting TB if the person you're enrolling has already got TB. So, you know, you've completely stuffed your trial. So it's it's a very good point, Simon. Thank you for raising it.
I know. I just wish we could, you know, go back and historically chest X-ray people. You know, it's always really frustrating when something like this comes up. You realise, yeah. Okay, so we'll do a few more. Whilst I'm looking at the chat, we can go down and do a few more of the questions in the chat, and then I'll flip back to the live questions. So we have a question from Rita Gossin. Is the BCG studied in the intravenous study given at the same CFU concentration as the intradermal immunisation, or is it less?
It's a really good question, Rita. Yeah, can you remember the answer to that, Helen? I have a feeling.
I think it's more. I think the intravenous might be up to 10 times more than was given intradermally.
Yeah, so my recollection is that in Sally Sharp's study it was less or the same, but that in the NIH study, Bob Cedar and Trish Dara's paper, it was more. That is my recollection. And of course, you're right, changing the dose confounds the result. Absolutely.
Yeah.
It's a good question.
Yeah, so it could, and it is true. I mean, it could be, part of it could be a dose effect. So if you gave 10 times the dose intradermally, would you? To be honest, I don't think it does. I think there really is something about the intravenous route, and you seem to get great delivery of the vaccine to the lung if you give it intravenously. So I genuinely, I don't believe it's just a dose effect. I
agree with that. And I think the issue there is that this is a replicating vaccine. So actually, dose is relatively unimportant in BCG. Certainly in the animal studies, it doesn't seem to matter so much what dose you give within a reasonable range because it replicates. Dose is much more important for vaccines that don't replicate.
Okay, thank you. So we have another question. So if exposure to environmental mycobacteria gives some level of immunity, why the immunisation of pre-exposed hosts with BCG does not increase the level of protection as if it was boosting immunity?
Yeah, so well, that's masking and blocking, isn't it? Paul, that's you. Or I can take it if you want.
Well, lots of discussion of this over the past 7 decades or something. Now, one of the things is to recognize that there are many different kinds of environmental mycobacteria. There are hundreds of them, and not all have been described by any means, and many people are exposed to these things. And they're immunologically different, and so just what overlap there may be in terms of their antigens with the antigens of BCG or M. tuberculosis. Every combination is out there. Difficult to study. There's been a lot of concentration on particular Mycobacterium intracellulare in the initial work. Why? I think interest in that organism started with studies of bovine tuberculosis in the USA, and so there happened to be those antigens, and they were used for testing US naval recruits for many years. There were those data, but you know, whether under some circumstances partial protection from exposure to one or another mycobacteria might be boosted by BCG, that might well be so. That, depending upon the extent of boosting, it may be very difficult to see.
Yeah, I think that's
not
right.
Easy, not easy to study. Probably all of us, I suspect all of us have actually been exposed to a variety of different environmental mycobacteria, and it's difficult to tease out the sensitivity which we have, and to do it on a large scale in a trial is a real challenge.
Yes, absolutely. I agree with that completely.
Sorry, I've just noticed the time. I'm having so much fun. I didn't realise that we've only got 3 minutes left. So I'm going to use chair's discretion and choose the last question to ask you. Lots of people are thanking you both, by the way, for the wonderful talk. So is there still a worldwide scarcity in BCG production and supplies? Has this negatively influenced policy around BCG use in recent years? And what does this mean for its continued use or for innovation in new vaccines going forward? So it's a really great question, I think.
It's a great question, and I agree. Thanks for picking that one, Helen. So, I mean, there are lots of good questions in the chat. So the simple answer is no, there is not a worldwide shortage of BCG at the moment. It is true that in the 20 years I've been doing clinical trials in this space, the number of global manufacturers making BCG has shrunk significantly. So when I started, there was a plant in Liverpool called Evans that manufactured BCG, but they don't anymore. They don't exist anymore, at least in this space. And I think that the number of manufacturers of BCG has more than halved in the last 20 years. So that's a problem. And it's a problem because there's no money to be made out of TB vaccines, certainly not making a vaccine that, as we've been discussing, is 100 years old. So that's a problem. We do, but it isn't. So what also happened was that a very big manufacturing plant in Copenhagen that made Danish BCG was bought by a different company, and that went offline for several years actually, but that is now back online, manufacturing, now known as AJ BCG, and is just the same as the old SSI BCG. So that's back in. And we also know that the Serum Institute in India are not only manufacture a significant amount of BCG, but are working with Stefan Kaufmann on that VPN vaccine I showed you. So we're actually committing to some manufacturing process development, which is decades overdue, to actually improve and more standardize the manufacturing process for BCG. So I think that's all very good news. I will just add one more thing, which is that there was a real concern in the TB community when the world thought that BCG might protect against COVID, that the global supplies of BCG might be rerouted to protect anxious people in the West against COVID, rather than protecting people with no data to support that at all, rather than protecting the vulnerable people who need it to protect against TB. I'm glad to say that hasn't happened. And as Paul said, happily, we now have very specific vaccines against COVID, and it's unlikely that BCG would get anywhere close to that the level of efficacy we see with those specific vaccines. But it is a cautionary note about getting too excited about the non-specific effects.
Okay, thank you both. We are out of time. Please do look briefly in the chat before we close out to see the adverts for the next public talks. But what a wonderful way to spend World TB Day and the 100-year anniversary vaccination of BCG. So thank you both very much. That was fantastic, and I hope you get a chance to look at the chat, at all the wonderful comments that you've got. Yeah, and thank you all for participating and being such an interactive audience to work with. So thank you.
Super. Thanks, Helen. Thank you for chairing so well. And thank you, Blakely, for doing all the behind the scenes organising.